Welcome back, my dear students. Now, in the previous section, I told to you about the different antioxidants. I am repeating a few antioxidants so that you can emphasize on this. These are very important antioxidants. Antioxidants can be enzymatic or non-enzymatic. So, we were looking at the enzymatic antioxidants, out of which the first antioxidant, which is enzymatic, is superoxide dismutase. I was telling in my previous lecture, O2 superoxide combines with one more superoxide and protons to form basically hydrogen peroxide plus oxygen. Hydrogen peroxide plus oxygen. These reactions need not be balanced. You need not know them also. So this is the action of superoxide dismutase. And where did you see this? This was happening even in the phagolysosome. When NADPH was being oxidized, oxygen was converted to superoxide. Superoxide was further converted to hydrogen peroxide. So let us look at where is its distribution. It is present in the cell cytoplasm. As I have told to you, the whole cell needs, everywhere it needs different antioxidants to prevent it from damage. If you look at the nucleus requires its antioxidants, the mitochondria require its antioxidants, the endoplasmic reticulum requires its oxidants, the lysosomes require, the peroxisomes require, each of these the cell cytoplasm requires. So SOD is mainly in the cytoplasm. Is there anywhere else? Yes, SOD is distributed both in the mitochondria and in the cytosol. Now in both these there is a slight difference. One is a copper containing enzyme, copper zinc enzyme, which is present in cell cytoplasm, and in the mitochondria it is a manganese containing enzyme. It catalyzes the dismutation of superoxide anion. There is also an extracellular form of superoxide dismutase which is present in plasma, lymph and synovial fluid. So we have it outside the cell also. So mitochondria, cell cytoplasm and outside also. This is superoxide dismutase. What is important about this enzyme is that it requires copper or it requires zinc or manganese. Now let us look at the next enzyme, enzymatic antioxidants, that is catalase. Now catalase is found principally in peroxisome and to a lesser extent in the cytosol and the microsomal fractions. So its main function of catalase is in the peroxisomes. A few is present in the endoplasmic reticulum or in the cytoplasm. Now what is its main function? In granulomatous cells like maybe macrophages, the enzyme protects the cell against its own respiratory burst. Remember, during the respiratory burst, hydrogen peroxide was formed. So we need a mechanism to protect these cells. This hydrogen peroxide is acted upon by catalase and it gets converted to water and oxygen. Again, I have not balanced the reaction. Please don't bother about it. What is important is its action. Catalase acts on hydrogen peroxide and converts it into a non-radical product like water or oxygen. That is important, catalase. Now, what else is important? It decomposes hydrogen peroxide to water and oxygen. As soon as the action of hydrogen peroxide is over, it is converted to hydrogen water and oxygen. Remember, the cell does not know how much of hydrogen peroxide is required to kill a particular thing. So if something extra is produced, we need to have a mechanism to defend it, and that is provided by catalase. Now let us look at the next enzymatic antioxidants, that is glutathione peroxidase. Glutathione peroxidase, the word peroxidase tells you that it also acts on hydrogen peroxide. Peroxidase means peroxide. Now it is present. Where is it present? It is present in the cytosol and mitochondria. So it has two isoenzymes, one of which is a selenium containing metalloenzyme. The other is a non-selenium containing. Why is it important? Selenium acts as an antioxidant. So it catalyzes the reduction of hydrogen peroxide and lipid peroxide by forming reduced glutathione. So to tell it simply, whether it is hydrogen peroxide or a lipid peroxide. I'm just showing it as a lipid peroxide. Now both these can be converted to the respective non-peroxide forms, maybe water or the non-peroxide form of lipid peroxide. But during this conversion, glutathione, which is in the reduced form GSH, that is how we show it, is converted to, two are converted to GSSG. We say that there is a disulfide bond being formed. I'll just repeat it here again. GSH, what happens, it gives its hydrogen, and two of them combine and form a cysteine molecule. Cysteine and cysteine combine to form GSSG. This is oxidized glutathione, whereas GSH is the reduced glutathione. So GSH is oxidized to its oxidized form GSSG. This is involved, glutathione peroxidase is involved, peroxidase. So it is involved in the peroxidation of hydrogen peroxide or the lipid peroxide to its respective non-reactive oxygen species. So let us go again. What will happen once this glutathione, which has been oxidized, how can this be regenerated? So we have another mechanism in the body by which GSSG is converted to glutathione back to its reduced form. I'll just show you this in separately. GSSG is converted back to GSH. So how is this done? This is done with the help of the enzyme glutathione reductase. Glutathione reductase. So what is required for this? For this we have an important thing that is required, and that is NADPH. Glutathione reductase requires NADPH. This is converted to NADP plus. So from where did this NADPH come? Please remember HMP shunt. So it came from glucose. Glucose was responsible for the formation of NADPH. NADPH, by the action of glutathione reductase, converts oxidized GSSG to reduced GSH or glutathione. This is one of the functions of NADPH. So let us go ahead with glutathione reductase is over, glutathione peroxidase, GPX is over, which is responsible for removal of hydrogen peroxide. We have catalase, we have superoxide dismutase, and their distribution in the various tissues. So let me talk a bit more about glutathione, which is an important antioxidant. Now this is the most abundant intracellular non-protein thiol, because thiol, why? It contains SH group. So a non-protein, remember glutathione is a tripeptide. So a non-protein thiol, most abundant within the cell is glutathione. It is mainly present in the cytosol and mitochondria and endoplasmic reticulum and nucleus. So wherever you see, glutathione is present, endoplasmic reticulum, but the concentration is less in various other parts, mainly in the mitochondria and the cytoplasm. Remember, glutathione is very much abundantly present in the RBC, especially in the cytosol. Remember, RBCs do not contain nucleus, they do not contain mitochondria. So in the cytoplasm, its concentration is very high within the cytoplasm of glutathione, and that helps in maintaining the RBC membrane integrity. So let us look at more about it. It functions as an antioxidant by scavenging free radicals and in detoxification of xenobiotics also. So the electron donor in the reaction of peroxides catalyzed by glutathione peroxidase, and it is converted to GSSG. It is reconverted back by glutathione reductase. So we have two enzymes involved in the metabolism of glutathione peroxidase and glutathione reductase. This is something which should be known to you, because here it is NADPH is there, whereas here it is acting as an antioxidant. So that is important. Now this finishes our enzymatic antioxidants. Now let us go further with the vitamin antioxidants, the non-enzymatic ones. Now one of the most important vitamin antioxidants is vitamin E. It is also called as alpha tocopherol. This is very widely distributed and it is a potent lipophilic antioxidant. It is present in cell membranes. It acts as a chain-breaking antioxidant and a free radical scavenger. So let us look at other vitamin antioxidants. That also includes beta-carotene. Beta-carotene is a precursor of vitamin A. It is present, carotene means it is present in carrots. It is highly present in all the yellow colored vegetables like including papaya, mango, etc. So it is beta-carotene is a precursor of vitamin A. It is an effective scavenger of alkoxyl and hydroperoxyl radicals. Vitamin C is another vitamin antioxidant, that is ascorbic acid. It works as a cellular aqueous phase antioxidant. So vitamin E was lipophilic and present in the lipid phase, whereas vitamin C works in the aqueous phase. It also reacts directly with superoxide or with hydroxyl ion as well as with lipid hydroperoxide. It acts as chain-breaking antioxidant by regenerating the reduced form of vitamin E, the alpha tocopherol. Let me just show it to you how it happens. So, if you look at the action of between vitamin E, lipid and the water, the aqueous phase, how these antioxidants interact with each other. So, let us look at the cell cytoplasm, and you have the cell membrane which is containing phospholipids. So, what happens? Now, the phospholipids are there. Most of the time, please remember, more the unsaturated fatty acid that is present in the cell membrane, more is its effect as a free radical, more is its chances of getting into lipid peroxidation. More the PUFA, more the lipid peroxidation. So unsaturated fatty acids, they undergo, first thing is they react with some free radical and the radical becomes non-radical product and oxygen, and it gets converted to a peroxy radical. Polyunsaturated fatty acid, lipid peroxide is formed. This lipid peroxide is highly reactive and it will undergo chain reaction. So what is there? This lipid peroxide should be terminated. We should terminate this chain reaction. So what do we have? We have to convert it into its OOH form. So what is required is tocopherol. This is, remember, this is still happening in the membrane itself, the membrane phospholipids. So tocopherol is converted to its radical form, but this can be reconverted back to its active form where it can scavenge free radicals by vitamin C. Vitamin C is present within the cytosol. Vitamin C is present in the cytosol, will react with tocopherol, and this gets converted to its own reduced form, and it regenerates vitamin E. It could be either even because of glutathione which is doing this, and they are regenerated back using NADPH. So basically we have seen that vitamin E acts as a chain breaking antioxidant. Vitamin C also can act as a chain breaking antioxidant because of its reaction with vitamin E. Now I go on to one concept, that is oxidative stress. As we saw till now, free radicals are being continuously produced. At the same time, there is a mechanism to prevent these free radicals from acting. So we have something which is happening in the body. One is free radicals, other is antioxidants. So what is oxidative stress? So in the normal cellular metabolism, there is a balance is between free radical production and the antioxidants. There is normally a balance, and so we are all healthy. Okay, so free radical generation and antioxidant production. But in sometimes, especially in certain clinical conditions, it has been found out that the free radical generation is more and the antioxidant capacity is less. The antioxidants are not able to scavenge out all these free radicals. The result is the free radicals are going to do their damage. Remember, they are highly reactive. They can damage the DNA. They can damage all the proteins, they can damage the enzymes, they can damage the lipids, they can bring about cell integrity itself can be damaged. So these free radicals have to be scavenged. So your antioxidant levels are low and free radicals are high, then it can result in a disease, and this is called it as oxidative stress. So this imbalance in the pro-oxidant or the free radical generation and the antioxidant level is known as oxidative stress. So are there any markers by which we can come to know whether a person is under oxidative stress or not? Free radicals have been implicated in all the diseases. So if you look at it at the cellular level, it is always because of free radicals that something is happening. If you want to look at DNA damage, then also it is because of free radicals. You look at cancer, free radicals. You look at arthritis, it is free radicals. You look at cardiovascular diseases, free radicals. At the molecular level, it is always because of free radicals any disease process is occurring. So let us look at the markers for oxidative stress. Are there any markers? One, we can have something which is damaged, which is released, which we can mark, or we can look at certain antioxidant levels itself within the blood. So which are the markers? Now one important marker is malone dialdehyde, which is a marker for lipid peroxidation. Though this is something which is not being used in routine clinical practices, research is going on whether to include this or not in different clinical lab settings. Maybe it may not be necessary because we all know that at the molecular level it is because of free radical generation and because of lipid peroxidation that a disease is occurring. So malone dialdehyde is a marker for lipid peroxidation. We have oxo-DG, what called as deoxyguanosine, 8-hydroxyl-2'-deoxyguanosine, which is a marker for DNA damage. Again, from an MCQ point of view, I am talking about all this. The protein carbonyl content is the marker for protein oxidation. Glutathione redox ratio, whether GSHG is more or GSH is more, the glutathione redox ratio is a measure of cellular redox state. Let us look at the clinical significance of different oxidative stresses and how they will help. We'll look at atherosclerosis. As I have told to you before, atherosclerosis is mainly because of low density lipoprotein, which is the bad cholesterol. Now LDL, can it always lead to bad things? No. This LDL is a lipoprotein. Remember, this lipoprotein is just to carry cholesterol. Now this lipoprotein, if it undergoes some damage, only then the macrophages will think it to be foreign and engulf it, and then it becomes bad. So what are the factors which cause a change in LDL, which we call as oxidized LDL? So oxidized LDL has come because of action of oxidants. So LDL are deposited under the endothelial cell and they undergo oxidized LDL by free radicals, and that attracts the macrophages, and macrophages engulf this oxidized LDL and they get converted to foam cells. There is one disease known as chronic granulomatous disease. What is this disease? Remember, in the phagolysosome, for the initial formation of superoxide, in this we require NADPH oxidase. NADPH reacts with oxygen to form superoxide. Now this is something which is necessary within the body. This is something which is necessary for the killing of a bacteria. Suppose superoxide was not formed, what would happen? Now what is that? That is known as chronic granulomatous disease. So in these people, NADPH oxidase is absent, and what will happen? Where is it absent? In the macrophages and neutrophils. So it leads to defective phagocytic function. And what is one more thing is happening is the bacteria are engulfed and they form a granuloma. They are not getting degraded because there is no superoxide, there is no hydrogen peroxide, there is no hypochlorous acid to bring about the lysis of the bacteria. So what will happen? There is this bacteria will remain within these WBCs or within these neutrophils, and it will form a granuloma, and slowly that is why the condition is known as chronic granulomatous disease. It leads to they are also susceptible to different microbial and fungal infections depending upon it is a catalase positive action or not. Chronic granulomatous disease. You will learn more about this in the later years. So right now just remember that it is NADPH oxidase that is deficient. So we go further, one important area, that is role in aging. Free radicals play a very important role in aging, and they are involved in neurodegenerative disorders like Parkinson's, Alzheimer's, dementia, multiple sclerosis. In all these conditions, there may be other things which may be involved, the genetic levels or something else, maybe some other reason may be there, some other protein misfolding may be happening, but all that is attributed because of increase in free radicals. So in many of these diseases, they give antioxidants, or they give fruits and vegetables which are rich in beta carotene, vitamin C, etc. So that is the reason why we have to take all these fruits and vegetables to increase the antioxidant levels within our body. Now I'll look at the carcinogenesis and treatment. Yes, free radicals mediate DNA damage that leads to mutation and finally malignancy. Free radicals are also involved in chronic and acute inflammation, ischemia, reperfusion injury, and anything and everything. Free radicals have been implicated. So with this, we come to the end of free radicals and the different antioxidants and their role in health and their role in disease. Thank you.